Namaste and welcome to a new episode of the Bharatvaarta podcast. Today we speak to two very accomplished professionals who come from investment banking and consulting background, but are now working in the field of cancer prognosis and diagnosis. They have created a path-breaking testing innovation, which can have deep impact not just on the healthcare sector, but on the society as a whole. Let me say hello to Anish Tripathi and Ashish Tripathi.
Thank you, Ashish. Pleasure to be here.
Yeah. Pleasure to host you. So before we get into the discussion, let me quickly introduce Ashish and Anish. So Ashish Tripathi is the co-founder and CEO of Tzar Labs and director of Epigeneres Biotech. Ashish has had 20 years plus experience in investment banking, corporate finance, and strategy, spanning Australia, Europe, US, Asia, and Middle East. Tzar Labs is in the business of development of cancer prognostic and diagnostic technologies and specializes in discovering, building, and commercializing innovative tests which can contribute to a clinically meaningful meaningful detection of cancer and management of cancer disease. Anish is a senior advisor and India head of Tzar Labs and also the CEO of Epigeneres Biotech. He brings in nearly 30 years of experience cutting across geographies and sectors like consulting, professional services, and manufacturing. Epigeneres is a leading biotech company that discovers, develops, and commercializes molecular medicines for the treatment and prevention of serious medical conditions like cancer and diseases of the old age. Thank you both for taking your time out of your busy schedule of Bharatvaarta, and we are really excited to talk about this wonderful innovation that you have brought. You're supposed to bring into the market very soon.
We're going to be. Thank you.
Thank you. So let's start with an introduction to the work done by Tzar Labs and Epigeneres so far. So perhaps if you can just talk about what areas you've been focusing on and what type of research. Ashish, maybe we can start with you and then we can come to an.
Sure. So essentially, Epigeneres was actually a molecular medicine company. In essence, what it was working on was creating immunotherapy drugs. So effectively drugs that act on specific genetic markers. And we were focused in the area of diseases of aging, particularly cancer. Now, while we were working on that, we found certain genetic markers that we were able to isolate from the blood, and these are transcriptomic markers on the basis of which we were able to actually develop a early diagnostic tool for cancer. So if I was to actually describe our business, we are essentially a molecular medicine and molecular diagnostic company, and our core area is in genetic breakthroughs that can actually help both sides of the business.
Right. Anish?
I would only add that we've, as of right now, we've just announced the breakthrough that we've had on the diagnostic side in cancer, and but it will lead to more developments and more breakthroughs both on the molecular medicine as well as the diagnostic side.
Right. And in fact, what we did for this episode was we crowdsourced some questions, especially from doctors, via social media, etc. So I think some of these details we will look at more closely in terms of what the diagnostic is all about.
Yeah.
But one curiosity question, and for both of you, I mean, investment banking, consulting, legal firms, from that to molecular medicine, how did that transition happen for both of you?
So look, the good thing is that all the people that are involved in this company have specific areas of specialization and certain competencies that we actually bring to the fore, right? Research is one thing, but the reality is that if you want to translate it into a business outcome, right, there are specific skills that the team must possess, and it's my fundamental view. It's not an individual that actually makes successful business. It's a team that works in a focused way to achieve a certain outcome. And from that perspective, there are certain skills that I bring to the fore. I have over 20 years of investment banking experience. Have worked around the world in US, UK, Australia, Middle East, India, and done very large transactions. So effectively, obviously, have competencies in who are the core investor bases that will actually look at investing in certain ventures. Similarly, if you look at Dada's background, right, he is a specialist in process engineering. And believe me that there is when you're trying to actually work on a tight timeline to achieve a outcome like this, where you want to launch this on a large scale to cover a population like India, at a fundamental level, you need to actually streamline the process. And to be very honest with you, his is a task that I'm glad he is doing and I'm not, because it's a difficult proposition. And similarly, we have, my father is a great scientist, and even at 80, coming up with fantastic research breakthroughs. And we have similar team members. So, the way I see it is that we've all got our skill sets, and we work in a very coordinated way to achieve certain outcomes, and that's what makes us successful.
Absolutely.
All I will add to that, Ashish, is that my father has been doing. He's a nanotechnologist. He's been doing research for the last 10 years. Ashish joined him on a full-time basis maybe around 5 years back, and I joined around 1.5 years back. And at least the way my father Ashish and I look at it is that this is a blessing from above. So it's we are custodians of this knowledge, and it required for such a significant breakthrough. It required an all hands on deck kind of a thing for the whole family, and this is really what we are doing to be able to bring like this something like this out, which is a testimony to Indian scientific talent. And we're just the faces. I mean, there are a lot of people behind the scenes who are working very hard, and we are here because it's a blessing that needs to be taken care of.
Right. No, great to know. So let's talk about the actual innovation itself. Ashish, if you can talk about how did the HRC test how did it get conceptualized? What was the research process? And how did we get here? How much time did it take? So if you can give some background on that.
So, the simple answer would be that this breakthrough actually was created over the last 4 years, right? But it would be an incomplete statement, because the reality is that there's a decade worth of experience and expertise that the team had made in molecular markers, right? Because we were basically creating drugs, that were working on these genetic markers, if you will, right? And my father is is someone who's worked for 20 years on nanotechnology based technologies that can increase bioavailability of molecular drugs. Right. So there was a lot of expertise that went into making this. I think in some of our interviews, both Dada and I have actually, noted certain specialists, right, who have done so much work in the area of primary research, right? Because without their work, it would have been very difficult for us to actually have been able to make that innovative leap that we did, and come out with this particular breakthrough. So, what I've realized, Ashish, is that a lot of people actually don't know how the HRC marker works. So if I may, let me just explain what HRC is, right? So when I take a blood sample, in essence, what we have created is a proprietary algorithm by which we can take transcriptomic markers and we can actually create this HRC scale for anyone from their blood sample, right? So if I was to take your blood sample or my blood sample, right, and I did an HRC test, it would give me a score anywhere between 0 and 60, right? And the way it works is that if you're healthy, you're in the 0 to 2 range. right? Now, there are certain inflammations that move this marker in the 2 to 6 territory. You're still healthy, but there are certain pressures that are forming on certain tissues and organs in your body, right? The most innovative stage, insightful range is actually 6 to 10, because what we have discovered is that this is the range when certain mutations are happening in certain organs in your body, and your tumor is starting to form, right? So understand that, for example, when you look at when when a tumor is formed, even the smallest tumors would be like, let's say a 1 gram tumor is a 1 cubic centimeter tumor. It has approximately 100 million cells of cancer, right? Now, it doesn't form in one day, right? It forms slowly over a period of time. What we are able to do is detect it before that tumor has formed. And not just that, we can also then, when on this marker, 10 to 20 is stage 1, 20 to 30 is stage 2, 30 to 40 is stage 3, and 40 and above is stage 4. So the interesting thing about this marker is that it is a very accurate screening test for cancer, because it actually tells you all the stages of cancer, right, from a blood marker, but it also tells you all the stages of non-cancer from the same blood marker, right? Which makes it a very powerful screening test. And once we found the markers, then it was a question of actually testing it on multiple types of cancer. We needed to do cell line studies, we needed to do animal studies, we did human studies, right? But we were able to then actually create a very accurate scale. And that's how this particular study got to a stage where we could do 1,000-patient study. 1,000-patient study is a fairly large study, right? And we were able to actually show on so many different types of cancer that this marker works extremely accurately. It can differentiate between cancer and non-cancer blood, but it actually can differentiate between the stages of all these different stages of cancer and non-cancer, which is what makes this a worthwhile discovery.
Right. Fascinating. So effectively, we are saying that it's like a stage 0 discovery. It's not the stage 1, but it's like a pre-stage 1 kind of a discovery, right? Which can be brought on.
Correct. So the way to look at it, it actually is the earliest. So there are a few things that we would claim that this is the earliest detection for cancer, because we can track it before tumor forms. This is the first prognostic test for cancer. So we can not only tell you that you don't have cancer, but we can rule out cancer risk for you for the next 1 year if you're within a safe zone, right? And not just this, we are able to actually, we haven't actually covered this in too many of our interviews, but we can even tell you what is the specific type of cancer and the primary organ and the primary disease that is growing. But that's going to come later, because there are other clinical tests that are being done.
Right.
Ashish, what I would like to add is that it's interesting you use the term stage 0, because stage 0, the way we define it, now stage 0 is not something which is unknown to the scientific community at large or oncologists. It's just that there was no diagnostic test to detect cancer at stage 0, which is really what we've been able to find. So stage 0 is when the underlying genetic mutations have occurred. The possibility of cancer stem cells starting to form has started, and then that will ultimately lead to cancer tumor cells, and then they coalesce together to form tumors. So that is stage 0, which we've been able to identify. So stage 0 is not a new thing that we have nomenclatured as such. It's been something which is known to oncologists, but practically speaking, oncologists have never been exposed to being able to diagnose something at stage 0, which is what I think with our test they will be able to see now.
Right. Yeah, I think that's a great explanation. This was actually one of the questions which we got as to from one of the doctors. So I think this kind of explains the process. Yeah. So Anish, talking about clinical trials, Ashish mentioned there have been 1,000 patients who have kind of undergone a trial. So how's that process going on right now? What are the other requirements which you need to fulfill before some kind of commercialization can start?
Okay, so the trial is over. It was CTRI registered trial. The trial is over, and based on that, we published our paper, which was published in a peer-reviewed, very reputed journal, Stem Cell Reviews and Reports (SCRR). And so that part is over. Now there are 2-3 things that we are working on. We are working on setting up our own lab in Mumbai, where we'll be able to roll this test out. There are certain regulatory approvals which are required. It's not very tedious or cumbersome, but we're working on those. And the reason it's not, there are certain procedures which we need to follow and we need to comply with, which we will do. The reason why it's not very tedious and cumbersome as compared to what people would normally expect with something to do with cancer, because there is from a regulatory perspective, there is an underlying unstated dictum, and that says that if you bring something new into the market and you claim something that you'll be able to do some benefit to subjects or patients, it's fine. We will look at it, but it should not do any harm. If it does any harm, if it has any potential to do harm, then the regulatory mechanism is very harsh, and rightly so. It should be, right? There are many more hoops that you have to jump through. Ours is a blood test. We have done a study and we made certain claims, right? So a blood test, the worst that can happen to you is you'll have a bruising on your arm, right? When there's a blood being withdrawn. The worst that can happen is that it may not work. So there is no harm that the blood test can do to you, which is why the regulatory system is less harsh. It is more lenient, and we do not expect our glide path towards regulatory approval to be problematic at all.
Right. So yeah, I guess the test being non-invasive is probably
it's a non-invasive test. Exactly.
Yeah.
Thank you.
Yeah. Right. Exactly. Okay. So Anish, you spoke about the setting up of a lab, etc. Do you need? Will this lab need any specific type of equipment? How quickly can you scale? Can you go from one lab to say 100 labs? What's the process for that looking like?
We are working on right now. So we of course need to roll out our own basic lab, and there is planning undergoing right now in terms of how we will expand and scale so that we can make it available to. We are obviously launching it in India first. We've also announced that it will be most competitively priced as compared to any other test, the same test in any other part of the world. And but in terms of how to scale, that's something which is work in process. What I can tell you is that our plan by the end of either this calendar year or financial year is to be able to set up enough capacity to do something like 1 lakh tests a day. That's the plan that we are working towards. Having said that, I will say, and COVID has taught us that India is such a massive country that any capacity that is set up is short always. So there will be more work to be done there, but those plans are underway right now.
Right. Yeah, even if the labs are few, there could always be a logistics type of a partnership with hospitals, and I think right. So, Ashish, one question for you in terms of the, I think we spoke about a term called stem cell research, right? And I think this has come out in the various literature, etc., which has been published by the company. So this question came from two different doctors both from Mumbai, Dr. Amit Thadani and Dr. Rajesh Bijlani. They wanted to know that is stem cell a misnomer here, or what is the relationship between the test and the concept of stem cell research?
So it's a very perceptive question. The fundamental, we have actually looked at the problem in a very different way, right? Most of our competition has actually been looking at tumor cells that can be traced in the blood, right? Whether you're looking at CTCs, whether you're looking at cell-free RNA, whether you're looking at ctDNA, and so on and so forth. We attacked the problem from a different way. We looked at a causative marker through the use of stem cells, right? And in essence, what we were thinking, what our process was, that there are certain mutations that will happen that lead to the cancer formation, right? And it has two fundamental benefits, right, with the approach used by our competitors. One, because it's a causative marker, right, it tends to be a lot more accurate. So long it's complex, but if you can crack it, it is a lot more accurate, right? Because it doesn't suffer from the same problems like false negatives, etc., that you will associate with the other process. The second thing is that it gives you the entire cross section of the disease, right? So it is actually, we are fundamentally getting a reading when you are a non-cancer patient. We are getting a reading for when you are an imminent cancer patient. We're getting a separate reading for when you are a cancer patient, right? Now, that's not what happens with the cancer-based approach, because all you see is tumor cells present or tumor cells absent. It's like a binary. Here, you're actually getting an empirical reading for every stage, right? And that, again, shows in our data when we have actually conducted, I mean, this was a blind study of 1,000 patients, and we got no false negatives, no false positives. But the answer to your fundamental question is that stem cells is very core to the breakthrough that we made. That said, the application is in oncology, and it is going to help oncologists, right? That's perhaps why, so you can have the basics can be any cell that you like, but at the end of the day, you need to understand how this is relating to a specific set of doctors. And we understand that our research is basically going to appeal to oncologists.
Right. So, Ashish, you spoke about competition. Are there any other tests which are taking the same approach anywhere else in the world, or is it like the first of its kind globally?
So this is the first of its kind, and one way to always look at these things is when you look at patents, right? So a breakthrough like this, you file patents in multiple countries. We've got patents in US, Europe, Japan, Korea, Canada, Australia, just to name a few, right? So we have filed patents, and we've realized that the core breakthrough that we have found has not been replicated by any other any other individual, or at least they've never filed any patents for it, right? As far as we're aware, we're the first people to have isolated these specific cells in peripheral blood, right? And and then identified these specific markers, right? So that is what we have done. That said, there are a number of liquid biopsy companies that have been trying to find markers in peripheral blood that can early diagnose cancer, right? There are very reputable companies from US in particular that have been working on this for quite some time. But as you will see from their research data and publications, they are nowhere close to the accuracy, nor do they have the same breadth of coverage of the number of cancers that we can cover, right, using our methodology. So there are certain very promising differences to our technology, our accuracy, and the ability of our method to compete, right? But it is a very unique breakthrough.
So Ashish, the way I would just simplistically put it is that the fundamental difference between what we've done, which is a global first, and what some of the competitors that people talk about are doing, is that their starting point is when cancer tumor cell formation has happened. Because then they're looking for either those CTCs themselves, they're also called circulating tumor cells, or they're looking for its fragments, which is DNA or RNA-based fragments. So their starting point is that. Our starting point is one or actually two steps before that, which is when the underlying genetic mutation has happened because of our focus on stem cells, right? And we were the first to be able to isolate those stem cells in the blood, as Ashish said. So our approach starts at least one, but actually two steps before what all the competitors are doing. So that's a simple difference between what we are doing and what the other companies are doing.
Right. Yeah. So Ashish, one follow-up question on this. You mentioned about patent filing in different countries, which is of course critical to the process of commercialization in such an innovation. But from a launch perspective, are you looking at other countries? I think, Anish mentioned India was the first one, but then wouldn't it be easier to scale it up in some other country than India, given the relative complexity?
So, it's early days. We've just published our data, and we've already had companies reach out to us from Europe, from Turkey, from Mexico, from Middle East, from Africa, right, to name a few. And the important thing to understand is that we are very proud of the fact that this breakthrough has been made by Indians, right? And as such, we are emotional about wanting to launch this in the Indian market first, right? And that is what we intend to do. But that said, this is look, cancer is a major issue for the world, right? There are 10 million people that pass away every year from this disease. It is going to help all Indians, but frankly, it's going to help all mankind. I mean, my wife is Australian American, and the reality is that if you look at the kind of pain and the kind of financial troubles this disease brings, not just to the patient but also to their families and to the society at large, this is a major problem. So our intent and the manner in which we are going about it, the reason we want to do formal clinical trials globally, etc., is because we envision a world where all of us should do this test once a year, right? And we will either catch the disease at stage 1 or before, right? But that said, there's always a place where you start, and for us, that is India.
Great. And the trial was about 1,000 patients. So there would have been a certain number of cancers you would have already identified in those 1,000 patients, right? While the universe could be much larger. I believe there are what 180 types of different cancers. Yeah. So
less than 200.
Less than 200, yeah. So, so going from the number you found in those 1,000 patients to, let's say, 200 or 180, are more tests needed for that, or is it just if you can prove it for 60 or 70 or 80, can you prove it for 180? I mean, how does that process work?
So, Ashish, look, the underlying markers that we are looking at, and since there's a scientific paper available, people know this, that the underlying markers that we are looking at, right, actually work across cancers. The headline that you would have heard is that in this blinded study, there are 25 different types of cancer. So it was a blind study where any cancer could be given to us. As it so happened, there were 25 different types of cancer given, right? All the large cancers were there, right? So you had breast cancer, lung cancer, prostate cancer, pancreatic cancer, colon cancer, right? Leukemia, right? All the large ones were there. But what fascinated the doctors is that it works on solid cancers, it works on blood cancers, it works on sarcomas, right? And now this is not a surprise to us because we had already done early studies, and we knew that it technically works, across all types of cancers, right? But the way we see it is that there will be larger tests done, right? Especially in the Western world and so on and so forth. The reality is that we know the underlying science, and therefore we are able to tell you that this is a common test for all cancers. And that is what makes it a powerful, and a non-invasive process to find it as well. So it has a huge application.
Right, absolutely. Great. So there's a question which came from a scientist and a researcher who actually did not want to give his name. He's on Twitter with a, let's say, a pseudo handle. His question was, would it be right to interpret the pluripotent VSCLs as the rogue cells that cause cancer? And if yes, then is there a potential cure for cancer by way of restricting the formation of these specific cells in the bone marrow?
So again, a very, very perceptive question, right? And you need to knowing what the nature of these cells and understanding how they work, right? It would be right to actually, there are certain reasons that this particular cell mutates and leads towards formation of cancer stem cells and therefore cancer, right? Frankly, there are many researchers. This is a cell that has been researched by 40 institutions around the world, right? And there are many institutes that are working on therapeutic use, right, of these cells, right? We ourselves are, on as a company, right, working on the Epigeneres side. We are working on ways that we can actually act on certain genetic markers, right, and therefore come up with drugs, right, that can lead to immunotherapy drugs, right, which can actually which can actually cure people more painlessly and earlier than current medicines that are available, right, to patients. That said, right, our core breakthrough, right, we are focused in what we have done. We have used the diagnostic applications, right, by these cells. And the point of the matter is that, when we actually get into the deeper analysis, this is not the only cell that we look at. We look at other cells as well, right? And we understand the correlation between these cells, which is why we are able to give very deep diagnostic information to oncologists, right? And the answer to your question is yes, there are brighter minds than us that are actually focused on that particular part of the problem, right? But we understand what we have done. We have made a diagnostic breakthrough, which will help a lot of oncologists in the treatment of these diseases, which are based on these molecular markers.
So can I just add, Ashish, one small thing to this at a philosophical level? The true cause of cancer is something which is not known, and there are people who are working on it, and hopefully we will find it in future. So what causes these cells, or what causes the underlying genetic mutation, is something which is not known. Even though there are studies which talk about high risk factors like smoking, carcinogenic elements, etc. But exactly what causes cancer to happen is something which is not known. What we have been able to do is to reach at the next best thing, which is whenever the underlying genetic mutation happens, if we can capture it through a test, you will be able to catch it at the earliest possible stage and take treatment and corrective action, so that the chances of survival, the cost of treatment, the time of treatment, all of that can go down. But what really causes that genetic mutation is something which hopefully we will discover in the future. But as of right now, I would say that nobody knows the answer to the question.
Right. But that said, if I can say one thing, there is a thought out there that it's your own body's cells that turn against you, right? And we are, based on our research, right, we are of that viewpoint as well, right? And to be very honest with you, which is why we believe that actually by attacking the problem in a different way, by actually looking at your own core cells rather than looking at the tumor cells, right, we have actually been able to, attack the problem in a totally different way and find a much more meaningful and something that is translational from a diagnostic perspective.
Right. Yeah, I think the concerned researcher probably asked the question, which was a one step further, which is to say that if the diagnostic can be easy, then can the cure also be easier in terms of the next step of the process? I think that's probably also it's a natural lead for your innovation, which kind of helps. Probably your own labs, but also others as well.
There are some experts who believe in this, by the way. It's not. There are experts who are working on this as well.
Right. Anish, Dr. Gautam Govitrikar from Pune wanted to know if you found any oral cancers in the sample that was clinical trial ran on, because he believes that could be a good first point of advocacy against gutkas or cigarettes, etc. if we can isolate the oral cancers. So did you find?
I don't immediately recall. Ash, was it there?
There was. In fact, one of the high risk patients that we found. Right. Yes. So as I told you that see, in a blinded study, you don't know who the high risk patients are. Right. You will only chance on them. right? And the reality is that there was a patient who was a smoker, right? And he showed signs on this HRC scale that he was imminent cancer, right? And because we were able to then do a follow-on study and say that this guy is about to get oral cancer, the good thing about oral cancer is that actually the biopsies are easier, right? Because the access is easier, right? And they were able to confirm that yes, he's at he's got the underlying mutations, right? But in answer to your question, it works on
All
it works on
cancers.
all cancers, so it works on oral cancer as well.
Right. There was a question from Dr. Amer Angnekar, who's a orthopedic surgeon from Indore. I think you have answered this question, but I should anyway read this out because kind of concerns which the doctors have raised. So his question was that in spine surgery, we come across a lot of met tumors which are often diagnosed late as secondary. And there's a lot of reliance on histopathology reports, which may have observer bias. So his point was that would this test be more accurate? I mean, clearly the answer is yes, but I think it's good to kind of address
That's a good
that.
question.
No, no,
Ashish, the only point I'll add to this is that this is a good question because it demonstrates that a lot of cancers get discovered by accident, by a radiology report that you wanted for some other part and you discover it, or in surgery like this doctor has said. So this is true that cancers get detected at stage 3, stage 4 many times by accident. And our test offers a genuine possibility of being able to capture it at the earliest possible stage 1 or earlier, right? And so hopefully if everyone is doing this test once in a year, we will not catch cancers at that later stage. I think that's the core message that I would like to give to Dr. Govind.
Right.
If I can just add to this is actually a very important question, right?
Yeah.
Because it's not just in the example that he's given, right? But primary unknown is a known phenomena by a lot of oncologists, right? Where you know that the person has cancer, right? And while the clinical trial report has been published in Stem Cell Review and Reports, you can actually read it. It actually gives you the methodology, but it also gives some very interesting case studies. I'd like to describe one particular case study because that will interest Dr. Govind, right? There was a particular patient, right, whose blood samples were given to us. Unbeknownst to us, they had gone in 3 days later for a surgery. When we got the samples, we accurately detected that this is a cancer patient and what the stage of cancer is, right? And of course, it's a blinded study, so we are going on with a number of other samples. Now, this particular patient, they removed her ovaries, they removed the uterus, they removed the fallopian tubes, at the end of which they realized that the CA125 was negative, right? So this is not ovarian cancer. Now, this is not something that happens in India. It happens all over the world, right? Like I say, to the good doctor's point, right? This is a primary unknown, right? And the proposition done by the doctors, the next steps described by the doctors was that they felt that this could be a Mendham cancer, and they wanted to do certain tests because obviously you need to have treatment strategies, right, for this individual. Now, the patient did come back and tell the CRO that they don't know what the primary is. The CRO asked us that can we help, right? And we were because we already had the blood samples, right? We were very quickly able to do the study. We were able to pinpoint that this person has appendix cancer, right, from a blood sample. Right. So incidentally, I have not spoken because I know you asked the question, what are the other clinical trials that we are doing? Please understand that there are other clinical trials that are in progress, right? Because the first clinical study was about finding out that the HRC is a good screening marker for cancer, right? But we realized at the end of the study that there are many other things that we are able to do which help oncologists, right? And so there is a clinical study on right now. But what we can tell you is that we will the studies will show, but we expect that we will be able to also tell you what is the primary organ, what is the primary disease. And there are a couple of other things as well, but that is the key primary aim of the study that is actually in progress, right? And if we can do that, the logic then becomes that not only can we detect, right, imminent cancer, we can also tell you which cancer and where it is growing, right? Straight from a blood test. Now that is a very meaningful result, right, for the individual, for the doctors, for the oncologists, and we believe that this technology then saves a lot more lives.
Right. Yeah, I think that is quite fascinating. I mean, you talk to a doctor for something else, but that could then essentially help you detect a cancer early. I think that would be a really phenomenal practical application of where we are.
But I'd like to say that that's a very intelligent question, and obviously takes a doctor to actually. So I like the format that you have chosen, where you are actually asking doctors questions, because you can definitely see that in the quality of the questions that are being asked.
Right. Anish, one last question from a doctor's perspective, and then I'll have a couple of my questions, which is, Dr. Bansal from Indore, who is my actually good friend from my school times, he wanted to know how easy or difficult is it to train people to conduct this test in labs, and can that be scaled easily? Because he feels that sometimes some of these complexities come in the diagnostic process itself from a commercialization standpoint.
Let me answer that question a bit indirectly, if I may. The blood processing techniques that we use are not humongously different from the way others do this work. So it's not that. Our uniqueness and innovation has been in where we have looked and what we have looked for. That's really why we've got our breakthrough results. Having said that, and you would have guessed it when I said that we are looking towards setting up a capacity to process a large number of samples. And given my background, I'm a mechanical engineer by qualification. So we are actually looking at automating processes. And if we are able to do that, then we will be able to scale up the testing massively. And then the dependence on individual skill will go down, which is there when you have a manually done process. So it's an interesting question, and we are working on it. Hopefully, we should be able to scale, because like Ashish said, our dream is for every person on earth, not just in India, to be able to get access to this test and do a simple test once in a year, and stand a chance to be able to catch, if you are unfortunate enough or destined to get cancer, to be able to catch it at the earliest possible stage. So we will have to scale it, and we're working on that.
Right. Great. So I think we covered a lot of medical queries. One question from my side.
Thank you. Very interesting questions, Ashish. Thank you.
Yeah. No, so my question, I mean, I probably know the answer, but it would be good to hear from you as to why was the test named HRC?
Ashish, go ahead.
Look, anyone that has and most of us know people who have suffered from cancer, and as have we, right, as a family, and we lost someone very, very important to us. My brother-in-law, and Himanshu Roy, was a highly respected top cop from Maharashtra, but was well known in many parts of the country. And he is perhaps one of the most inspirational people that I have known. And unfortunately, he fought fourth stage cancer for 2 years. And, we lost him, 3 years back. And he was an individual who was, one of the most poignant things that he had actually, comments that he had made is that, this disease is unfortunately becoming a disease for the rich, right? And because if you look at the cost of medication, and, it is almost unaffordable, right? In many ways. Now, this is the reason why we were extremely passionate about trying to do something about this disease. It's not very well known, but Himanshu Dada and my sister Bhavnadi have done a lot for helping the poor, right, who are suffering from this disease. And, which is why in the ethos of the company itself, it's not just about actually making this particular technology available, because the reality is that, as Dada said, that we're working on mechanisms so that we can actually have coverage all across India, right? But the important thing is, from the ethos of the company, we also want to keep the pricing low, because we want it to be an affordable thing for all Indians. And our view is that, if everyone is doing this, right, you will catch the disease at stage 1 or stage 0, right, when it is infinitely more curable. And our core aim is, we need to make this into an early stage disease from a late stage disease. You do that and you will save a lot of lives. And that is the core aim. And the reality is that we named it HRC because we've named it in memory of our brother-in-law, Himanshu Roy. So it stands for Himanshu Roy Cancer Test.
Right. Yeah. We had actually hosted Bhavna Roy, sister of Anish and Ashish, on a previous episode of Bharatvaarta in connection with her book on Dharma, where she very passionately spoke about social good and how it's the responsibility of all of us to really look at solving problems around us in the society. And I would encourage the viewers and listeners to refer to that episode as well. It was not, of course, not related to this discovery, but a larger picture of how we all can contribute to society in our own different ways. So this was a very fascinating conversation, Anish and Ashish. I really learned a lot myself. And I really hope, I think a lot of people are excited, like when we posted this on social media. A lot of people, I think, and as you said, a lot of us are personally impacted by it in our families, right? So, I have seen in my family as well. So we can almost always, almost kind of jump with joy that, okay, if this commercializes, and then, that can save really a lot of social issues around us in terms of financial issues, in terms of family fabric and so on. So really excited about this. I wish you all the very best for an early commercialization and success. And thank you for taking your time out for this conversation.
Thank you, Ashish. Thank you. My compliments to you for the format and the kind of and the quality of questions that you got. It really was inspiring, and the kind of questions that this has raised in the minds of people is very very nice. Thank you.
And I'd like to add that you also have a wonderful name.
yeah, of course, always good to. I do have a double A though in my Ashish. So.
Thankfully, neither of us is triple A rated.
That's for the test, obviously. So let's hope it is a triple A test very soon. Thank you.
Thank you.
Thank you so much.
Thank you for tuning in to this episode of the Bharatvaarta podcast. If you want to see more content like this, then don't forget to subscribe to our channel. We started Bharatvaarta to facilitate long-form discussions on politics, policy, and culture. We don't necessarily endorse anything that was said in this episode. If you wish to offer us feedback, do reach out to us on social media. We are at Bharatvaarta on Facebook, Twitter, and Instagram. You could also get in touch with us on our website, www.bharatvaarta.in. The links are in the description below. Until next time, stay safe, take care, and Jai Hind.